Okay, so we left off with carbs and fats the other day, and now here we are. We're going to look at our other two macromolecules, proteins and nucleic acids. So, proteins. You think of hamburgers, mussels, they're everywhere. They are important in structural support, they're important in storage, transport, cellular communication, movement, and defense against foreign substances. The amino acid is the smallest subunit of a protein. Here is an example of your basic amino acid. You'll note the R group at the top. That is what varies between amino acids. You do not have to know all the different types of amino acids, but you should know that that is where they vary. Here's some of your non-polar amino acids. These are the ones that do not like water. If you look at the side groups, there's no hydroxyls, nothing that will interact, nothing polar. On the other hand, here's your polar amino acids. Look at these side groups that are highlighted in white. You'll see a lot of hydroxyl groups. That's what you're going to expect to interact with water. The peptide bond links the two amino acids. This should be a review for you. This peptide bond is formed, once again, by dehydration synthesis, just like we saw the other day. Loss of a water allows the two amino acids to bond to one another. The bond actually occurs between the amine group of one of the amino acids and the carboxyl group of the other. So let's look at this diagram. You can see that your amino group is in blue and your acid carboxyl group is in red. I told you that these functional groups will be very important to you. I was not lying. If you look at the bottom, you can see the newly formed dipeptide, di being 2, and there we have our peptide bond. We see the carboxyl group has lost its hydroxyl in order to bond to the nitrogen, who has lost a couple of hydrogens in order to make the bond with carbon. Polypeptide is a polymer, many units of amino acids. This can range from just a few amino acids to 1,000 amino acids in length. Protein formation, a whole nother story unto itself. There are different levels of structure which will determine the shape and function of each and every protein. The primary structure is the sequence of amino acids in a single chain, and this is determined by your DNA and RNA. Here's a picture of primary structure. You just see it's just the amino acids all lined up. But we're not done. We have a secondary structure that helps in the formation of these proteins, and this is folding due to the hydrogen bonds that are within the chain, and they will form between different amino acids. There are two different secondary structures. One is called the alpha helix. The other is the beta pleated sheet. And here in your here's a diagram at the top right-hand corner. You can see the beta pleated sheet. It's actually pleated, not just very. It is what it is. It is what it says it is. And at the bottom, you can see the alpha helix, which is a loopy helix. And you can see the hydrogen bonds in both are shown by the dotted lines. Tertiary structure is the next level of structure, and this is folding due to the R group interactions. It can consist of hydrogen bonds between different R groups of the protein. It also can consist of disulfide bonds. So here's where our sulfur or sulfhydryl group is important. And here's a diagram of tertiary structure. You can see how it becomes a little more mishmashy. Finally, we have quaternary structure, the fourth level of structure. And this is where you put together 2 or more polypeptide chains. Hemoglobin is a classic example of quaternary structure. And here 4 chains or 4 polypeptides are held together by an iron group. And these diagrams are of different quaternary structure molecules. The one in the middle is collagen. The one on the far right is your hemoglobin. We have to consider denaturation at this point. This is the loss of protein conformation and thus the loss of function. And this is really important when we talk about proteins. We're going to be seeing this again and again, that once the tertiary structure is lost, the protein cannot function. Let's take a break and look at that in real time. So now it's time for fun with paper towels. This is my favorite way of explaining protein conformation. So I'm taking an ordinary roll of lovely Seekonk High School paper towels and ripping off a large piece. This is an example of our primary structure. Notice how it's linear, strand, and just, humor me and think of it as being a bunch of amino acids that are all bonded together by peptide bonds. Now, we're not done. We have to go through secondary structure. Secondary structure is when we get the alpha helix thing happening. So if I begin to twist like so, and it's a little humid, you can see how it's getting a little curly-cuey, and it's not behaving, but that's okay. You get the idea, right? So that would be my alpha helix. The other type of secondary structure is the beta pleated sheet, and that simply would be folding, like so, like pleats. Okay? And guess what? You can have both in one protein. Okay? You don't have to have one or the other. They're not mutually exclusive. So here's my lovely secondary structure, but we're not done yet. We have to go to our third level of structure, tertiary structure, and this is due to those R groups interacting with one another. And you'll get disulfide bonds here, and then you get this big clumpy thing. This is what the functional protein looks like. Okay, and it has a very unique shape. It has a little cleft in here, it's got this, it's very unique, very special, and you need to know that. Now, tertiary structure is not done. We also talked about quaternary, where we have two or more lumps put together. And I just happen to have a lovely, lovely lump over here, which is also quite unique. And they come together, and here is our final product. Just a note, you don't have to have quaternary structure. Okay, only certain proteins require it. Many can function all by themselves in tertiary structure. And that's fun with paper towels. But wait, there's more. I almost forgot. We have to talk about denaturation. What is that exactly? Please don't say it's killing the protein, because AP really hates that. Denaturation is actually the destruction of the protein, and it can happen through heat, pH, a number of things. And so let's look at our little friend, our tertiary structure. Denaturation is breaking of those bonds. Now imagine if the disulfide bonds were to break, the hydrogen bonds were to break, what will happen to our tertiary structure? It'll eventually unwind, and there's no longer its normal form. Without being in its normal form, it can't function. And as we go through this year, you're going to see exactly how that plays out. And so now we really are done with fun with paper towel. One final note with protein molecules is regards to chaperonins, and their job is exactly what their name implies. So always look at those root words. They chaperone or help the protein to fold properly. So they help in making that tertiary structure. And now, last but not least, let's look at nucleic acids, the final macromolecule group. Nucleic acids are very important because they store and transmit all the hereditary information that you need to be you. They carry energy as well. Examples are DNA, RNA, and ATP. They are polymers that are called polynucleotides. The structure of a nucleic acid, it's composed of a nucleotide, that's the monomer, the single subunit. Nucleotide is composed of a nitrogenous base, a pentose sugar, pentose meaning five-sided, and a phosphate group. Here is a diagram of a sample nucleotide. So we have the phosphate group in orange, the deoxyribose sugar in this case, the pentose sugar is in blue. And the base, which we know that there are 4 different kinds in DNA, is shown here in green. There are 2 types of nucleotides, and you need to know them, and you need to know the difference between them. First is the pyrimidine, which is a single ring nucleotide. And I remember that, and this may help you, is pyrimidine sounds like primary, which means 1. The other type of nucleotide structure is a purine, and that one has 2 rings. In DNA, the sugar is deoxyribose, but in RNA, the sugar is ribose. And so DNA gets its name from deoxyribose, RNA gets its name from ribose. Here are pictures of the different bases at the top. This is stolen from your book, actually. And at the top are the 3 single ring bases, and the bottom are the 2 double ring bases. Why are there 3 single ring bases, you might ask? I want you to remember the difference between DNA and RNA. There's 1 base that's not the same. Now, we have to look at dehydration synthesis again. And this is in the making of a nucleic acid. We join 2 nucleotides by covalent bonds, and they form between the hydroxyl group on the 3' carbon of one nucleotide and the phosphate that's on the 5' carbon of the next. And this is called a phosphodiester bond. And these links will create a backbone of sugar phosphate units, and they will have the bases, your nitrogenous bases, as appendages sticking out. The 3' and 5', I want you to recall from when we looked at the carbohydrates, is the number of the carbon. And this is going to be really important when we look at DNA replication later this year. This is a nucleoside tri-phosphate, and that is the form that a nucleotide takes when it is not part of a nucleic acid. So it has, notice it has 3 phosphates. This should remind you of ATP, adenosine triphosphate, same family of molecules. And what do you suppose must happen in order for this nucleoside triphosphate to attach to a chain? If you're thinking that it must lose something in order to make a bond, you are right. It actually has to lose 2 phosphates so that it can make its bond with the adjacent nucleotide. DNA, I'm sure you remember, forms a double helix. And A, adenine, always binds with T, thymine. Cytosine C always binds with guanine G. This is due to hydrogen bonding between the bases. They line up that way because they like to form those hydrogen bonds. And also, if you look at your book, you'll see that in these pairs, one is a purine, one is a pyrimidine. And this helps them to fit in the double helix. So now if you look at this diagram, we have our double-stranded DNA. And if you look at the very outside of both, we have the phosphate groups. They're highlighted in a beigey yellow color. Those are all your phosphates, and they are attached to all your blue sugars. And note that they are on the outside. On the inside is where we see our bases, and they are color-coded here for your convenience. And A is in green, and notice that it is a purine. It has 2. And T is a pyrimidine, and so it's only 1 ring. So notice that the distance between the sugars, and now let's look down below to where we have G and C. C in this case is the pyrimidine in a yellow, and G in orange is your purine. They make it an equal distance, and this is, and you can see the hydrogen bonds in between. There are 2 hydrogen bonds between adenine and thymine, and 3 between guanine and cytosine. And this is the only way the double helix can be formed, so that it's equidistant. They're happily making their hydrogen bonds. And let's go to the next slide. So surprise, that was the last slide. I was surprised too. Anyhow, don't forget, you can go back over this as many times as you need to so that you feel comfortable with the information. Don't forget, your text is a great resource, and I do have animations linked to your site if you want to utilize those as well. See you next time.